What do you usually have for people, attendees?
It varies, anywhere between 20 to 60. It just kind of depends on the week, what's happening.
Okay.
Good afternoon, everybody. We'll get started here at about 12:30. Okay, good afternoon, everybody. Welcome to the Quantitative Seminar. Thank you for joining us. We're excited to have Sarah Converse as our speaker this week. I'd first like to begin with a land acknowledgement. The University of Washington acknowledges the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within the Suquamish, Tulalip, and Muckleshoot nations. So as I mentioned, Sarah Converse is our speaker this week. Sarah is the unit leader of the Washington Cooperative Fish and Wildlife Research Unit and an associate professor in the School of Environmental and Forest Sciences and the School of Aquatic and Fishery Sciences at the University of Washington. Her research focuses on the development and application of methods to understand the functioning of populations, especially small and declining populations, and to improve their management. She works across a broad variety of taxa: terrestrial birds, seabirds, marine mammals, amphibians, reptiles, and fish. Her research projects tend to reflect two primary themes. One, development and application of quantitative methods in population ecology, particularly for small and declining populations. And two, development and application of decision analytic methods to inform management of populations. So everyone is muted right now, but if you have any questions during the talk, Sarah has agreed to take questions during her talk. You can submit those questions through the chat box. And then at the end of the talk, we'll have time for a Q&A where, in addition to the question box, you can also use the raise your hand feature at that time. So without any more delay, let me turn things over to our speaker, Sarah.
Thank you, Andy, and thanks for the invitation to do this. I was reflecting on the fact that 4 years ago today, I was packing up my car for the trip across the country, and got here and have been enjoying Quant Sem ever since. And so I'm glad to finally get to present. And I'm really excited to talk about the work that I'm talking about today, which is about demographic synchrony, and specifically synchrony in seabird survival. And I want to start by mentioning that this project is very much a team project. The way that this came about, a former postdoc, Martina Caden, decided to become a teacher, which is really cool. And she's actually teaching now in Stockholm. But it was going to mean that this work that we were working on probably wasn't going to come to fruition. And so what we decided to do as a lab was all pitch in together and work on this as a group project. So it's been a lot of fun. I think we've all learned a lot about it, about different things. Everyone's learning something new. So even though I'm the one presenting here, this is very much a group effort. And we're getting to the point where I think we've got something that we're feeling pretty good about writing up. And this has been a good, this seminar has been a good prompt for us to move things along. So it's been great. So let's see. I'm not getting my slides to forward. Let me try that again. I am going to try this mode because I couldn't seem to get them to move forward in full screen mode. There we go. There we go. Perfect. Okay. All right. So I'm going to talk today about synchrony in populations, demographic synchrony. And I'll start by talking a little bit about how synchrony arises and why it's significant, why it's important for us to think about. And then I'm going to introduce you to our friend, the common murre. And then since this is QuantSem, I'm going to spend most of my time talking about the model and some preliminary results. And then I'll talk at the end kind of briefly about some conclusions and things that we're thinking about doing for next steps. Okay, so demographic synchrony. So, so throughout the talk today, the setup that I'm thinking about is a spatially structured population. So what I mean by that is you've got a population and there's several different subpopulations that are spaced out. They've got some spatial structure to them. And you know, these are often referred to as metapopulations if there's some movement between them. So if those populations, those subpopulations are asynchronous, you get a pattern like in the upper right, in the plot in the upper right. So all of the populations growing, this is population growth rate. Over time they're growing, but they're growing completely independent of each other. So they're not synchronous. If you have synchrony, you get a pattern like what is on the bottom right, where the population growth rates are highly correlated across the subpopulations. So the significance of this is that the degree of synchrony tells us something about the scale of the factors that are driving population dynamics. So to get the pattern that we have in the upper right, you have to have factors that are driving that growth rate that are operating primarily at that more localized scale, at the subpopulation scale. Whereas to get a pattern like you have in the lower right, you have to have some factor driving that growth rate that's at the larger whole population scale, or I'm going to refer to it today as the global scale, the scale of the entire population. So it tells us something about what's driving population dynamics in a case like this. The other important point about it is that synchrony has important effects on conservation of species and the provisioning of ecosystem services, the things that we get out of ecosystems that we value. For example, if this was a fishery, the population on the lower right was a fishery, we would have, across a very large space, we would have really bad years and no population sort of making up for that in other places. So what this looks like in terms of implications for conservation is basically that populations that are highly spatially structured populations that are highly synchronous are at greater risk generally for decline and potentially extinction. So here the plot on the left is showing you two sets of subpopulations and the number of those subpopulations that are extant in a given year, where the black lines indicate an asynchronous, where the subpopulations are asynchronous, and the red indicates a situation where the subpopulations are synchronous. So they have, in both cases, the same average growth rate, the same variance, the same movement rates. The only difference is whether they have synchrony, whether those up years and down years are synced up, or not. And so you can see that what we get with Synchrony is we get a greater decline in the number of extant subpopulations over a given time frame. So basically what's happening is that it's a bad year for everyone all at once. They can't help each other out. They can't make up for it. And so you get greater and faster declines in the number of subpopulations. And eventually that can lead to global extinction of the population. And then on the right, what you see there is you see the effect of movement. So this is what we call the rescue effect, where you can see that things aren't quite so bad for the synchronous populations when they can help each other out, but they're still relatively bad compared to the asynchronous populations that can really help each other out, right? If it's a bad year in one, it's a good year in the other, there's a relatively high movement rate, and you get this really strong rescue effect. So Synchrony, it has important implications for conservation, what we get out of ecosystems. The next thing I want to do is, turn our attention to this particular case study that I want to talk about today, and that is the common murre in the Baltic Sea. So common murres are a circumpolar distributed alcid. They're the same species that you'll see here in Washington on the outer coast or in Puget Sound and in the Salish Sea. So they're sort of the typical seabird, just like prosolarids and gavids and other seabirds where you have this slow life history. So birds have delayed breeding. Murres start breeding, common murres start breeding around 4 to 5 years of age, and they raise only up to 1 chick per year. And then significantly for our purposes today is that they nest in these colonies on islands. So you can think of each island as a subpopulation in a spatially structured population. Okay, so just as a note, a few years ago, we worked on a paper where we looked at murres on one island, one colony in the Baltic. And what we found was that the survival in those individuals was linked to sprat. So sprat are a common forage fish, important in the Baltic. And we found that the survival of murres was positively associated with abundance and biomass of sprat. So that was at one colony. So what our interest in then was saying, okay, let's scale up and see what kind of effects this might have at a larger scale. Okay, so basically this gets us to our research questions. We have two. The first one really is just, is the variation in survival of these birds driven by large or small scale variation in the Baltic? So are they synchronous, driven by large scale variation, or are they asynchronous, and so driven by small scale variation? And then our second question was, okay, can we figure out, depending on the answer to the first question, can we figure out what might be driving those larger sort of small scale variation in survival by looking at predictors, covariates, environmental variables that might be driving survival? Okay, so a little bit about the data. I'll talk a little bit more about this, but on the right, what you have is the Stockholm archipelago. So Stockholm is kind of in the center of the map approximately. And we've got 10 different islands, 10 different colonies that we're going to be looking at. You can see that there's 2 fairly far to the north, 3 that are kind of in the center there, and 5 that are farther to the south. And I'll also note that one of those 3 that are kind of in the middle there, Kalkin, is the place where we had worked on the single population work. And so for variable periods of time, but overall between 1995 and 2017, there were efforts to band birds, recapture birds, and resite birds on these 10 islands. So we have this big mark recapture data set, and we want to use this to ask questions about synchrony. So that's all good, but the reality was that there were several kind of big challenges with this data set. First is that we had multiple capture types. We had to account for that. The second was that we had a suggestion of transients when we looked at contingency tables, goodness of fit test for this this data set. We had a suggestion that there's transients, and the idea of transients in a population, the way it shows up, manifests itself in a mark recapture data set, is that individuals have a different probability of being seen again, depending on whether they were just captured for the first time or whether they have previously been captured. So basically the idea is that some proportion of those individuals that you're seeing for the first time, you're actually not going to see again because they're just moving through their transient in the population. And we would expect that kind of transience to be age related because again, these birds don't settle down and start breeding until they're a little bit older. And prior to that, they're moving around some. So we probably had something going on where we have this transience, but our age is imperfectly observed. So we had to account for that. And then the last thing is we've got 10 islands and we know there are movements between them. So we have potentially 100 different inter-island movement probabilities that we could model. Modeling 100 different parameters seemed inefficient. So we had to come up with a different way to handle that. Okay, so getting into the model. So we'll have a series of slides that sort of dig into the model because this is QuantSem and this is what we do. So this is a typical multi-state mark recapture model with just a couple of little tweaks to it. So the observation model, this is sort of typical how we would think about the observation model for these kind of data. So we think about our observations as coming from a categorical distribution that's dependent on Z. So Z is the state, the true state of the animal I at given time T. And it's a function also of the animal I and the time T. So things like whether the age of the individual, what year it is, all of those kinds of things. And there are 15 different observation types. You can be captured at sites 1 through 8. Those are sites where there was only efforts to recapture individuals, physically recapture individuals. And then at sites 9 and 10, they had field readable bands on birds. So there were both efforts to physically recapture birds, but also to recite birds. So we can have a physical capture at sites 9 through 10. We could have just a recite only at sites 9 through 10, or in a given year, you could have both a recapture and a recite at sites 9 through 10. The last of the 15 observation types is not seen. So we basically have to model a recapture probability and a recite probability. I'm going to mention, and I'm going to talk more about age in just a second here, but pretty much everything that we do through this model is going to be modeled as age varying. So I'm going to have generally 2 groups of birds, those that are less than 4 years old and those that are 4 years old or greater, which were 4 years old or greater, we're basically assuming our breeders or our adult birds. So I'm not going to always, in fact, I'm generally not going to show the age variation in the model explicitly just because it became pretty messy to show both age and time and parameter type and so on. So just sort of across the board, we're going to have age varying parameters. So we have these 2 parameters, a recapture rate and a recite rate. We didn't have very many recites, so we actually just modeled a mean recite rate. Recaptures, we had a lot more, and we included a random effect of site by time. So we could account for spatial and temporal variation in the recapture effort and the recapture rate. Okay, so a second part of the observation model that I want to talk about is the age observation. So as I mentioned, we have this situation where if we just if we just looked at the birds that are apparent adults, and I'll talk about what that means in a second, we would see that we have this suggestion of transience. So basically, when the birds are banded, their ages are recorded as either a 1, which is a young a year, so a chick, a bird that was hatched that year, or it's recorded as a 2, which is a greater than young of year. So it's a bird that's at least 1 year old or older, and you can't age it once they're 1 year old or older, you can't age it more finely than that. But all of those, birds that are greater than a year old or greater are not actually breeders. Some of them are still subadults, they're moving around. And the likely result of that is if you treat those individuals that were given age class 2 at banding as the same, you're going to have this sort of transience effect show up, birds that seem to be behaving differently than other birds. So what we did is we treated the true age class of the individuals as latent, and we have 5 age classes: young of the year, 1-year-olds, 2-year-olds, 3-year-olds, and 4+. And again, we're assuming those 4+ birds are breeders, are adults. And so the age class model, basically, if you think about this matrix down here, if you're given a age grouping at first capture of 1, you're a young of the year, so you're in this top row of this matrix, you're definitely a young of the year, you're definitely age class 1. So the probability of being age class 1 is 1. We know you're absolutely age class 1. But if you're given an age grouping of 2 at first capture, you're definitely not a young of the year, but you could be 1-year-old, 2-year-old, 3-year-old, or 4+. And we can treat this as latent, and we actually do have the ability to estimate it because of variation within these age classes in the demographic parameters. And I'll show you that when we get to the results. And this is the basis then for all of the age modeling that we do, where what we're generally going to do is group the animals that are 4 years and older and group the animals that are 3 years and younger, so that we have basically subadults and adults. All right, so that's kind of the observation data. So now let's actually get into the process model itself. So again, this is a very sort of typical multi-state mark recapture model where you have 11 different possible states. And the probability that you're in a given state in time t is a function of this big array that draws on your state in the previous time period, individual effects, time effects. So your states, your true states there are 11 true states. You could be on any of the 1 through 10 islands. So that's the first 10 states. And the 11th state is dead. And so basically what this big matrix or array looks like, we just look at it for a given individual I at a given time t, is you basically have this 11 by 11 array, matrix. I'm not showing you the whole thing, but you can think of for example, to stay in state 1, you have to do 2 things. You have to survive and you have to stay in state 1. And this is the psi parameter that we think about as movement parameter. So this is movement from site 1 to site 1. So you stay, or you can survive and you can move to site 2 and so on. And then of course you have to die to get into the last state, the dead state. So that holds across all of our different, all of our 10 different sites. But of course, what we want to do is then break down these parameters into a movement process and into a survival process. So let's talk about that. So first, I want to talk about the movement. So again, age varying like other things. The first thing we did is we said, we're going to break it up into a Bernoulli process for staying home or moving. So you can either stay where you were, or you can move. You can stay in the island that you were at last year, or you can move to a new island. And then conditional on moving, the probability of moving from island S to island R between any two islands is a function of the distance between them. It's also a function of this island-specific random effect, which is included to account for this sort of fundamental attractiveness of an island. And you'll see the results of that in a little bit, but let's look at it more closely first. So first, you can think of this probability of staying home. So moving from island S to island S, we've got sort of this mean probability of staying home. And then we've got this random effect of island, which again is this sort of island attractiveness. And this is a property of the island. It influences whether you stay home, and given that you leave, it influences where you might go. And so then if we think about if you do leave, you've got one of 9 choices, right? 10 total islands. You've decided to leave where you were, so there's 9 places that you can go. And so what we do is you can think of this right then as a big, which of those you go to is going to be a big multinomial probability. And so what we do is we account for the fact that you didn't stay home. Then given that you didn't stay home, we have this multinomial logit where your probability of moving is a function of the distance between the islands, where you started and where you ended up, the island attractiveness, the attractiveness of the island that you might go to, and again, then you have this sort of island attractiveness effect, where this is something that influences both whether you might stay, and if you leave, where you might go. So that's the movement. So then we get into survival, and this is where we start getting into the issue or the idea of synchrony. So again, we're going to model it as age varying for subadults versus adults. And our survival rate for a given island at a given time is going to be described by an intercept, the effect of some covariates I'm going to talk about in a minute, and then 2 random terms. One of these random terms is common across all islands. So this is what we'll call the global variance. Okay, so it's global in the sense that it influences all of the islands, all of the colonies. And then the other random term we have is a site by time or an island by time random effect. So this is basically the local variation. And so this is going to be influenced individually by a local scale variance parameter. So I want to, just because we spent a lot of time thinking about this and agonizing over this, and we learned some things that were kind of interesting, I want to dig into this part of the model here, this local parameter, just a little bit and talk a little bit about that. So we basically, over the sort of period of time when we were working on this, we sort of entertained 3 different options. And you can think of them as very much analogous to fixed effects, random effects, and a model of the mean. So we could have said that this island and time specific deviate for survival rates came from a distribution that was controlled by an island specific variance parameter. Each island has its own variance parameter. They're estimated separately. They're not linked together. You can think of those as sort of analogous to fixed effects, but rather than for the mean, for the variance. And then option 2 is a random effect. So the idea here is that each island has its own variance term again, but those variance terms themselves come from a hyperdistribution. So this would be, this sigma parameter here is basically the variance of the variances. And then the last option was to say each one of these island by time deviates is coming from the same distribution. So basically, they're each moving around, but there's only one variance term that's controlling that. So the range over which they can move is common across islands, but whether it's a good year or a bad year at any given island is independent. So we would have loved to, we started trying fixed effects, and for reasons I'll show you in a minute, that wasn't viable. Then we tried random effects, and that sort of worked, but didn't work great. And then what we ended up doing was deciding to use this sort of model of the mean. So every island moves independently in terms of this, this deviate, but they're all being controlled by a single parameter. So the range over which they can move basically is common. Okay, so now covariates. So we said that our first question is, are these populations synchronous or asynchronous? And our second question is, can we understand why? So we looked at a few different covariates. The first is the North Atlantic Oscillation. Of course, the Baltic is not the North Atlantic, but there have been some suggestions of some relationships between what's going on in the Baltic and the North Atlantic. So we used the North Atlantic Oscillation. We used a model, we fit a model with both a year effect and a year lag effect. We also went back and looked at spread abundance. Spread abundance was something that was very important when we had looked at that individual island, the island of Kalkin. So we wanted to look at that. And both of these two are Baltic scale. So they influence all of the islands, or they're modeled as influencing all of the islands the same. The island scale variation that we looked at was the sea surface temperature estimated near given islands, again for year T, and then a lag effect. We also looked at the island protection status. So this is not time varying, but it could influence some island to island variation overall. So those were the covariates we looked at. And then last, I just want to make the point that what we did is we to think about this issue of synchrony, we calculated this interclass correlation coefficient, which provides a measure of synchrony. And so basically what that is it's just the global variation in survival over the sum of the global and local. So as this ICC measure gets close to 1, it indicates that that global variation is dominating, and that indicates synchrony. If it gets close to 0, that indicates that this local variation is dominating, and it indicates asynchrony. Okay, so just to be thorough, I thought I would just throw up the priors that we used. They're all kind of pretty standard, vague priors. We put the prior on the mean, recap, and recite rates on the probability scale. We gave those a sort of flat beta prior. And we also, to model that variation and recapture probability over space and time, we used a half normal with a standard deviation of 10. Our latent age, we gave that sort of a vague prior with Dirichlet 1111, so basically sort of equal prior probability of being in each of those age classes, given that you were, when you were first captured, you were greater than a young of the year animal. And then movement, so we have our movement parameters. So again, for the mean movement, we put the probability on the, we put the prior on the probability scale with, again, a beta 1,1. Our sort of distance covariate, which is beta psi, we gave that a normal with a standard deviation of 5 with the variance. So this is this island attractiveness effect. This is the sigma psi parameter. That's the island attractiveness. We gave that a half normal. And then for survival, to finish it up, sort of mean survival effect. Again, we put that mean survival, we put the prior on the probability scale, beta 1,1. Similar with these covariates, so the effect of things like SPRAT and NAO and all of this kind of stuff, we gave that a normal prior. And then we gave half normal priors to these two variance parameters. Okay, so data and results. So these are the data. Here are again our 10 different subpopulations. And you can see the capture numbers over time for the period 1995 to 2017. And the thing I just really want to highlight here is how, one, some islands had absolutely no effort and no captures in many years. And the number of captures was quite low for some islands in some years. And this is really kind of the moral of the story in terms of why we had to use that sort of simpler kind of model of the mean on our local scale variance. So every island has its own deviate from, but this from the same parameter, the same distribution with a common variance term for that local variance. We just really couldn't fit a model with more structure to it because of these big sort of missing pieces where we just didn't have any data. It didn't work very well at all. Certainly the fixed effects didn't work well at all because there's just no information to estimate those effects for many number of years. Okay, so let's get into results now. So here are, I'm not going to go into results on the detection probabilities. I'm going to start with the latent age class. So again, as I mentioned, because we do see such big age differences in survival and movement, this actually allowed us to, there's information in the data on latent age class. So we're actually able to take those individuals that when we first capture them, all we know is that they're greater than young of the year, and we're able to classify those in terms of the probability that they're members of these different age classes. So ages, age 1, 2, and 3, these are our subadults, and they get lumped in with our young of the year. And then age 4, these are our adults, and they get their own rates for pretty much, or their own parameters for pretty much everything in the model. So we are able to estimate those things. Okay, cool, movement. So this is super cool. I'm excited about this plot, and the lab put this together, and it's really cool. So I just want to take a few minutes to talk about this. So to orient you, each of these 4 subplots is looking at the probability of moving from one of the islands on the rows to one of the islands on the column. So if you're in year T, you're here in one of the rows, what's the probability in year T plus 1 that you end up in one of these columns? And we're doing this first for the probability of staying. These 2 on the top are the probability of staying, and the 2 on the bottom are the probability of going. So we wanted to show these on different plots because the probabilities are so different that the differences don't show up unless you kind of isolate them onto these separate staying and going. Okay, so let's look at this. So we'll start by looking at adults. So this is the probability of staying. And what this is telling us is that no matter where you start from, if you're an adult, your probability of staying where you are is basically 95% or greater. If you're an adult, you're just not leaving. And this is exactly what we expect from seabirds. They have very high breeding site fidelity. So we contrast this with subadults, where your probability of moving, or sorry, your probability of staying is much different. And now here's where this island attractiveness comes in. So the only thing that's allowing these to differ is this island attractiveness. Okay, so what you see is that some islands have yellow. Those are the most attractive islands. The green are kind of moderately attractive, and the blue are the least attractive islands. So basically, if I was at Calkin in year T, I am probably going to, even if I'm a subadult, I'm going to probably be at Calkin in year T+1. Okay, so that's the probability of staying. The probability of going down here in the lower left for adults, we just said, right, that the probability of adults staying is really, really high. And so the probability of adults going, of course, is very, very low. And you see that all of these rates for adults are really, really low. So not as interesting. What's cool is this plot over here in the lower right. So this is the probability of going for subadults, right? So what's going on here? All right, so if you think of from and to, right, from are our rows, to are our columns, what's going on here is two things. We see the effect of distance and we see the effect of island attractiveness. So the islands are organized from north to south. So what you get is two islands up at the top that are grouped together, three islands here that are kind of grouped together in space, and then five islands down here that are pretty closely grouped together in space. And you can see the effect of distance a little bit with sort of higher probabilities within those groupings. But the other thing that you see that's cool is the effect of island attractiveness. So let's look at Bådskär. Apologies to anyone who is Swedish or speaks Swedish. We're going to call this Bådskär. So up here, we saw that Bådskär is pretty attractive, right? It's got a pretty high probability for subadults of staying. It also has, if you're coming to Bådskär from somewhere else, the probability is pretty high that you would end up at Bådskär. So it's something about Bådskär that makes it more attractive, and so birds like to go there. Similarly with Calkin, something attractive about Calkin, and so birds like to go there. So the way we are sort of thinking about this as a lab is you've heard your whole life that it's not about the destination, it's about the journey. It turns out that for common murres in the Baltic, it is about the destination. So they care about where they're going, and certain places are just better places to be. Okay, I see that I have a question. Has there ever been any satellite tracking studies to verify any of this? That's a great question, Nicole. And I wish Martina was here to say more. I don't know of any. She is doing some light logger tracking, but that's an effort to understand sort of wintering movements at kind of a different scale. I don't know of any satellite tracking to verify any of it. Of course, what we do see are birds that are banded on one island showing up on another, but it does seem like it would be cool to be able to do some more tracking. Satellite tracking is tough with birds because of the size required of the technology. These aren't enormous birds, but yeah, but I don't know anything more than that. If you're very interested, I'm certainly able to get you in touch with Martina. Okay, so that's movement. So now let's go to survival. So the first thing that we're going to look at are the effect of the coefficients. And I guess the takeaway here, the punchline is that it's all very disappointing. We're very excited to look at these coefficients, but most of them either don't seem to have a strong effect, or the effect doesn't make a lot of sense to us. So I mentioned before that when we looked at Calkin by itself, originally, we saw this strong positive effect of spread abundance. In this model, we're seeing a negative effect of spread abundance that's actually relatively strong. And I guess our only interpretation of that at this point is we don't know what's going on. We're continuing to try to dig into it. We can recreate the positive effect if we allow Calkin to have its own parameter for the effect of spread abundance. We can recreate what we saw in the past, at least some. Obviously, the model structure is a little bit different, but we don't know why. And it sort of maybe suggests that there's more spatial structuring in sprat themselves, which actually makes sense. And again, what happens with so many of these forage fish populations, we just don't have the kind of small scale data that we would like to have. And so we're treating sprat as something that exists sort of at the global scale, but maybe it doesn't. Maybe it actually matters quite a lot what's going on in any given year in terms of where sprat are distributed. We have this positive effect of NAO lagged, and I think we need to think more about that, whether we think that that makes sense or whether it's a weird artifact or something. Yeah, we don't totally understand why we would be getting a lagged positive effect of NAO lagged. So we need to ask some more questions about that. I saw a question that just came in from Caitlin. Sorry if I missed this. Do you have any ideas about what makes certain islands more attractive than others? Great question. We would love to model some things. There's a variety of things. How many people visit? Some of the islands seem to be visited more by you. Some of you may be familiar with the fact that American mink are invasive in the Baltic and are found in the Stockholm archipelago. There might be something about eagles and whether there are eagles that are hunting in the area. There might be something about the size of the colony, the habitat, but we don't have any, at this point, we don't have any good covariates that help us to explain that. So that's why we're just treating it as sort of a random effect, but it's definitely something that we would, that would be really interesting to look into more. Okay, so now this is survival. This is a model with none of the covariates, and then this is a model that we just sort of threw all of the covariates into. Kind of the takeaway here is that adults, as you would expect, have high survival, relatively non-variable. Subadults are going to have higher survival, relatively variable. And you don't really see a lot of change when you add covariates or not, because generally they're just having relatively small effects on the whole. Okay, and then last thing is this interclass correlation coefficient. So the idea here again is that if the larger scale variation is controlling the survival rates, you're going to see high interclass correlation coefficients closer to 1. If more local scale drivers are influencing survival, you're going to see interclass correlation closer to 0. And so the takeaway here, let's start by looking at this model without any covariates in it, is that in general, it looks like these populations are relatively synchronous. Now you can see there's a lot of uncertainty, but that sort of mean estimate is someplace above 0.75 or so. What we should see then is if we add covariates that explain variation at one scale or another, it should change the NAO. So for example, if we have a lot of variation in that global scale, and then we're able to explain a lot of that variation with a parameter, with a coefficient, with a covariate, what we should see is that ICC shifts to being more dominated by local scale variation, and so the population should look more asynchronous. So for example, what you would say if NAO was important, if we don't consider NAO, the population looks like it's very synchronous, but as soon as we account for the NAO, then we see greater asynchrony. So it's asynchronous contingent on having accounted for the effect of NAO. That's what we'd like to see. we don't really see it very well. Again, none of these covariates seem to have a big impact on what we were seeing. I just have, I think, one slide left, and then I will go to more questions. So the takeaway here is that the survival of these birds in the Stockholm archipelago is driven primarily by these large scale forces, but what exactly those forces are, we are not really able to say at this point, given the small number of covariates that we've looked at so far. What it does suggest, though, is that this degree of synchrony suggests that there is going to be a greater sensitivity to large-scale changes in the Baltic, and those large-scale changes are of a lot of interest. The Baltic states are working really hard on things like recovering cod stocks, reducing nutrient inputs into the Baltic, and so on. Large-scale changes may have an influence, a meaningful influence on these birds because of this degree of synchrony. And then the last thing I'll mention is that we would like to examine the synchrony more at the island cluster scale, because we do actually have these island clusters in north, mid, and south, and the plot over on the right is just sort of an initial effort to look at whether we saw really strong patterns in synchrony if we look at the individual sort of clusters of islands. It might be that this level of synchrony is driven by island cluster as much as anything. So we're going to dig more into that. And then we're also going to try to continue to look for some covariates, and one of the first ones that we might look at is herring. All right, I want to just thank Morten Fredrickson, who's a collaborator on this from Aarhus University, one of the co-advisors on Martina's work, and then sort of the support for the work getting to the point where it is with Martina's and others' efforts. I want to acknowledge the co-op unit and the Swedish Museum of Natural History. And that is all I have. So.
Thank you, Sarah.
Yeah, I can answer. Should I answer the questions in the question thing here?
Well, I'd like to remind our audience that if they have any more questions, that they can enter them into the chat box, or alternatively, if you'd like, you can use the raise your hand feature, and we can selectively unmute you to ask Sarah your questions. But I think we'll start with a question from Maya.
Okay.
Maya, you should be able to unmute yourself.
Sure, can you hear me?
Yep.
Yep.
Great, awesome. So I had actually entered what I put in the chat right before you showed your last figure, but this is really interesting work. I'm super interested in connectivity and spatial questions too. And one of the things I was thinking about is wondering how your movement setup it might be confounded with your sort of local random effect, that mean effect you modeled. And what before you showed that figure, what I was thinking is, if you have set up your connectivity or movement based on the attractiveness of an island, what you're really saying is this island is going to get a subsidy every year of individuals, which in a bad year globally would effectively mask any synchronicity that might be shared among islands, simply because that nice island is getting a subsidy. However, in the results you showed, it seemed like everything was trending towards more synchronicity anyway, so that might be a moot point. But I guess the kind of secondary question I was thinking, I might have missed this, I don't really know that much about birds, but I'm curious about if you have any thoughts about how the kind of density dependence might play into this. I don't know if they're breeding and staying on one island, or if they breed and then those individuals are just as likely to move somewhere else, but that I've found is a major problem for our fishery models. So yeah, that was a bunch of words.
Great, no, thank you, that's great. So let me just quickly address the first question about effect of movement. You're right, and sort of early on in the talk, I showed sort of, you know, how movement can, a lot of movement can dampen the sort of negative effects of synchrony, but it dampens them even more when you have asynchrony, because basically bad year here, good year there, place where it's a good year rescues the place where it's a bad year. And so you get sort of that dampening. What we saw here was that other than subadults, birds don't move a lot. Some of the subadults move a bit, but those movements tend to be pretty localized, so within the island cluster than across island cluster. And of course, yeah, again, there is this attractiveness effect. But we can still see synchrony even, or sort of synchrony or asynchrony even when we do have movement. The movement isn't huge. And so, again, this makes us want to look into this sort of island cluster kind of effect a little bit as well. But yeah, it's a great question. Second question, density dependence. Yeah, density dependence. We haven't tried to take that on. We don't know really what the mechanism is. It's a little harder for me to believe that that density dependence would be influencing this population through survival. It seems more likely that it might be influencing it through reproduction. That's just my sense as an ecologist of what might go on here. So we haven't tried to account for it or model it. So we are just looking at these sort of, you know, observed survival effects over time, and sort of how they're driven by environmental conditions, rather than sort of these within population feedbacks. It's an interesting question. I'll have to think about that more. Thank you.
Okay, I think Ray was next with a question. Ray, you should be able to unmute yourself.
Okay. Can you hear me?
Yep.
Yes.
Yeah. Okay. Yeah, I wanted to point out that there's a paper that will be out any week now in conservation biology by Chris Free. It's a postdoc at with Olaf Jensen, where he put together as many data sets as he could find on the abundance of forage fish and their predators, and found a few more cases where the coefficient was negative than positive, but not most of it was no strong effects. But you know, I mean, I guess the question I posed online was, would you agree that if there had been a strong positive coefficient, this would have been all over the bird news? That but if there's a negative coefficient, they're going to say, oh well, you know, it's just, I think there's a really strong attention bias when it comes to to these things.
Yeah, that's a great. Oh, so a lot of thoughts about that. Well, so one thing I'll say is that we published a positive effect of sprat, and it was in Ecology and Evolution. That was where we got it accepted. So there wasn't a press release. Yeah, definitely need that paper. That's definitely going to be cited in our paper. So yeah, let me know when that's out, or if you can share something at this point. That will be really great to see. So one of the things I think about with this is that when we look at biomass or abundance, there's a couple things going on there. Right, we're not really accounting for distribution. We're not really accounting for not only distribution during the breeding season, but distribution during the winter, which can be really important. We're not really getting at those sort of how the birds are changing with respect to the populations. The other thing that matters too is, you know, there can be other effects. So for example, bigger fish aren't necessarily better. So if there are differences, you have your early age classes of a given fish growing faster, that can influence, you know, the degree to which they're the right size to feed yourself and feed your chicks and so on. So I think it is. I mean, I totally agree. It's a lot more complicated than just abundance. And so that's how we're going to write the paper.
Just
Yeah.
a couple follow-ups. I mean, one of the things, if you look at the Curie 1/3 for the birds paper, is, you know, what they really showed is it's really the local abundance that it probably is what matters. And so there was one example in Alaska where there were 3 colonies. They were all pretty close, but they had local abundance, and the one that had high local abundance had higher reproductive success. But at the management scale, they're all seeing the same managed population. You know, so you can see a local abundance effect, but it may not reflect anything that has to do with the actual the actual management. And just finally, we've been looking at the relationship between Peruvian anchoveta and the 3 main marine birds there, and we found a negative coefficient on the abundance of anchoveta, but a pretty positive effect of the amount of fishing pressure. Okay, the actual fishing effort. And the explanation that we've come to, which is I think people would agree, is it's the act of fishing, particularly near colonies, that breaks up the schools of the prey and makes foraging for the birds less efficient. So it doesn't really matter how many anchoveta there are in Peru, it's how many concentrated schools there are within the range of the foraging distance from the colonies. So it's not an abundant. So you might want to look at, and if you have any way of measuring that issue. I don't know anything about these species, so I don't know whether that's an issue. But Charlotte Boyd worked with us, and that was sort of her interpretation. It was this disturbing the high concentration schools that really made the foraging less efficient.
Okay, so you got a negative effect of fishing concentration on whatever survival or something of the birds.
Well, a negative effect of the total abundance of anchoveta, but anchoveta, but also a negative effect of more fishing was bad for the birds. That was the point.
Yeah. One of the things that's really cool, interesting, and yeah, I'm a little bit familiar with that work that you've done there. And one of the things that's frustrating is that for a seabird biologist is that many of the forage populations that we care about are not as closely, the data streams on them are just not as good as those that are more significant commercially. And so, yeah, those sort of small scale or very fine scale monitoring efforts, they don't exist. And so that's a challenge. I mean, we're thinking about that with pigeon guillemots and Puget Sound as well. What we think are important for them are not herring, where you have maybe some better data, but they're things that we don't have really much monitoring data at all on them. So that's also one of the challenges here. Yeah, great, thank you. That's really, really useful stuff.
Well, it is past 1:30 at this point in time, so I know a lot of folks have to leave us. Sarah, thank you for the great talk. It was really interesting.
Great.
And thank you all for joining us. If you are able, we hope to see you all again next week, where our speaker will be Christina Morissette from Utah State University and Henry's Fork Foundation. She'll be giving a talk optimizing multi-stakeholder watershed management in Idaho's Henry's Fork. So thanks again, Sarah, and we hope to see you next week, and have a great